Bonjour tout le monde. Bonjour tout le monde. Good afternoon, everybody, and welcome to the Amazing Brain Talks. We're very excited to see so many people here, and I think you're going to have a wonderful afternoon. This event is organized by Healthy Brains, Healthy Lives, a large McGill initiative that I will talk about later, and by Brain Canada. My name is Alan Evans. I'm the Scientific Director of Healthy Brains for Healthy Lives and a Professor of Neurology and Psychiatry at McGill University. I will be your MC today, and I would like to begin by acknowledging our presence on Indigenous land. We are meeting today on land which has long served as a site of a meeting and exchange amongst Indigenous people, including the Haudenosaunee and the Anishinaabe nations. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which people of the world now gather. We have a great lineup of speakers serving brain health researchers, and people with lived experience will share their knowledge and their stories with you. We'll cover a wide variety of topics, including the effects of cannabis on the developing brain, forecasting brain health with aging, the linguistic brain, and our experience of living with obsessive-compulsive disorder. I'm going to give a brief introduction of some of the activities that are going on that inform what we'll be discussing today, and then we'll go into a series of lectures. So, Healthy Brains, Healthy Lives. Before Healthy Brains, Healthy Lives started, we had a lot of activity in brain research at McGill, starting at the Montreal Neurological Institute, which was established in 1934 by Walter Penfield's seminal work on mapping the brain by direct cortical stimulation in the surgery. Herbert Jasper developed EEG at the MNI in the 1940s. EEG is a technique to map the spatio-temporal activity in the brain at a millisecond time base. It's a very, very fine time resolution. Brenda Milner, who many of you will know, Brenda built the field of brain-based psychology, now termed cognitive neuroscience. It was originally neuropsychology. And Brenda is still here today, causing trouble. Spends lots of time. I spend lots of time with Brenda. We actually watch Manchester City playing football all the time. She's a very, very avid supporter of Manchester City. And on the right, we have Bill Findell, who is a former director of the Montreal Neurological Institute, the MNI. And he was a very early proponent of what we call red veins, which is the basis for the technique that we now call functional MRI, where we use magnetic resonance to observe the living human brain. So there's Penfield getting his start, poking the brain and seeing if it elicits some sort of response. And I came in 1984, sorry. It feels like that at times. I was minding my own business in South Wales in the UK playing rugby, and best job I ever had was as a deck chair attendant at Barry Island. And then I met a Canadian that changed my life completely, and I came to Montreal in 1984 and got involved with brain activation studies in the early 80s. So here's an example of how the brain is able to react to external stimuli. What you're seeing in the middle there is just a rotating checkerboard, and you see how the brain, the back of the brain, which is the visual cortex, don't know why that stopped, how the visual cortex responds just to the orientation of that checkerboard. So we're looking inside the living human brain to see how that very subtle brain function is taking place. On the left, you can see where the brain responds when it's processing language, particularly verbs, the difference between nouns and verbs. That's where that is instantiated in the brain. On the right, you see that blob? That blob is where the brain responds to pain. We took some unsuspecting McGill students and put them in the scanner and put a thermistor on their forearm and raised the temperature. That's where the brain responded to increased temperature. So that's how you can map the various activities in the human brain. What you're seeing on the right is what Penfield did when he establish what parts of the brain are responsible for what functions. And that's what we look like to our brain. So this idea of brain mapping is not just for function. We also can use it to study disease or structure. So what you're looking at here, this is an image of an MRI of multiple sclerosis. And you see this here, that's a multiple sclerosis, sclerosing plaque lesion in one patient. Well, what if we do this across hundreds of patients? 425 patients, we get something on the right. The green stuff is what we call the probabilistic distribution of where you're going to find multiple sclerosis lesions in the brain. The red stuff is just the fluid ventricles in the middle of the brain. So think about that. What you've got there now is a huge amount of data from over 400 patients. And if we gave 400 people a drug for MS and 400 not a drug, we can find out what parts of the brain respond to that drug intervention. So if the green stuff gets bigger or smaller, that's the drug intervention. So you're collapsing vast amounts of information into something which is a very easy, digestible image. But it's all quantitative. It's not glorified neurophotography. This is quantitative analysis of the brain. Now, HBHL is predicated on the idea that we have to study machinery, the mechanisms and the circuits in the brain. When you look here, a traditional approach might be to look at a set of genes, and they might be associated with different diseases, disease 1, disease 2, Alzheimer's disease, Parkinson's disease. But the problem is that many genes are associated with many diseases. So there's what we call pleiotropy, a lot of mixture there. So what we really need to do is to look at the mechanisms. These genes code for circuits and mechanisms. And then those mechanisms appear in different diseases. But now we are understanding how the brain works, or how the pathophysiology in the brain is taking place. And we can then design techniques to interfere with that machinery if we need to. So it's a much more mechanistic, circuit-based approach to understanding how the brain works. Don't get worried. I'm not going to go through all of this in detail. This is just to give you a sense of HBHL's breadth. Many, many researchers who are all studying the brain, looking at connections and wiring in the brain, in normal brain, in psychiatric disorders, neurological disorders, and how do these circuitries break down. So it's a very quantitative approach. We apply these techniques to study many diseases, obviously things like Alzheimer's disease and Parkinson's disease. We're always trying to understand how does the machinery break down in a quantitative way. So this is my life, trying to maintain sanity with 240 principal investigators and over 1,000 highly qualified personnel involved with HBHL. We have vast amounts of data. What are we going to do with all of this data? Well, we have to develop infrastructure, computational techniques and strategies to reduce all of that data to something the clinician can use. In essence, we're just trying to build the Star Trek tricorder, where everything about you is in that box. Now, we may have big computers on the walls and distributed across the country, but conceptually, that's what we're trying to do, to make all this data available to the clinician at the bedside. We're not there yet. We will get there over the next 20, 30 years. But conceptually, what we're doing is no different from Star Trek. So we have computers that we have bound together across the country, across Canada, supercomputers, the cloud. We use all of those techniques to allow us to analyze these vast amounts of data. We have many international colleagues and international projects where they use supercomputers across the US, across Europe, and all of that now is accessible to us and to our international colleagues. This just gives you a sense of that. This, what we call C-Brain, which is this portal to the computing infrastructure around the world, is now in 32 different countries, now over 2,000 users. So it's now a global industry. It's no longer a cottage industry where one researcher, one student, and half a secretary are working on one hypothesis. It's now industrial strength, large-scale data acquisition and data processing, where many people can get access to those data and beat this data to death, take lots of new ideas and explore the same large amounts of data to extract information in a way that was hitherto impossible. So we have seen a sea change in how science is conducted. Very large scale, very open, and global in scope. Just in our own setting within Canada, we're very pleased to have Brain Canada represented here by Viviane Poupon, CEO of Brain Canada. Brain Canada supported the creation of the Canadian Open Neuroscience Platform. This allows us to take all of this data that we've been talking about and making it accessible to researchers across Canada. An example of that would be the Pan-Canadian Alzheimer's Disease Network. You have 340 researchers across the country, all connected through this common infrastructure, all asking biological questions of large import, whether it's Alzheimer's disease or frontotemporal dementia, vascular dementia, various forms of dementia. We have to tease all of this apart and understand the differential mechanisms of this disease versus that disease. Another example is the UK Biobank. Some of you might have heard of the UK Biobank. The British spent 15 years developing this plan where they collected data from 500,000 people. They made that data available to everybody around the world, and we are one of the beneficiaries of that largesse from the UK. But it's just one example of large amounts of data being put out there for the world to get access to. Another example that we're involved in is a long-term collaboration with Katrin Amunds. Katrin is the head of the European Human Brain Project, and her lab and my lab came together to explore a data set and build a data set that we call the Big Brain. And I'll explain why we call it the Big Brain in a moment, and explore artificial intelligence and large-scale compute approaches to understanding brain structure. So this is the Big Brain. And to give you an idea, this is the resolution, the smallest thing that we can see in the Big Brain, is on the order of 20 microns. MRI, that you're all familiar with, and I've shown you examples, has a resolution of about a millimeter. 20 microns is 50 times smaller than a millimeter. You do that in 3 dimensions. So it's 50 times 50 times 50 times smaller, and therefore the data set for the whole brain is 50 times 50 times 50 times larger. That's 125,000. So this single Big Brain is the equivalent of 125,000 MRIs. That's why we call it the Big Brain. Of course, we published this back in 2013 when that was big. It's 10 years later. It's now small. We are now working towards going to 1 micron, which is rather small. What you're looking at here is the structure of the hippocampus. The hippocampus is the structure in your brain which underpins memory. Everything that we've understood about memory, starting with Brenda Milner, who I mentioned before, lies in that structure. And we can now explore the basis of human memory in a way that was hitherto impossible. I'll close with just some comments about the globalization that I've already referred to. The Global Brain Consortium. This is an organization that its primary motivation is democratization of brain research for low and middle income countries. In the past, low and middle income countries around the world were a source of data. They were not involved in the research enterprise at all. Their data was taken, studied, researched, and published from the West. Now, with the rise of the computational infrastructure that I've described, they can now get access to not only their own data, but everybody else's data around the world and ask questions which are relevant to them. So it's very much about democratization of global brain research. We have projects which look at neuroscience of early adversity, stress of normal living, biomarkers of dementia, Parkinson's disease, child and adolescent brain development, COVID approaches. So there's a lot of studies that are going on and making use of this infrastructure to study a wide range of biological and societal challenges. Just as one example, in the UK, Faisal Mushtaq is looking at over 20,000 children who come from an immigrant population. So they have a very different societal experience than the people who were born, who are indigenous to that region. So their environment affects their brain development. All the things that I've just been described, to look at behavioral differences, social care, education, housing, police stress to the population, environmental challenges. All of these things affect their development and their societal engagement. So all the infrastructures that I've been described allow people like Faisal Mushtaq to explore the genetics, the cognitive behavior of children growing up in indigenous populations, so immigrant populations. So there's a lot of work that goes on. These are the leaders of the Global Brain Consortium, working really hard, looking at the big brain with popcorn. These are the people who are going to be speaking today. I won't go into detail here because you have your programs. But I think you'll find that the exploration of various themes today will catch your interest, and I'm looking forward to a very exciting day with all of these speakers.